So, good afternoon, everyone. Welcome to Session 1, Emerging Contaminants in Aquatic Ecosystems. I am Xinghui Xia from Beijing Normal University of China, and Professor Gisela Umbuzeiro from the State University of Campinas of Brazil, and I will co-chair this session. There are 1 keynote presentation and 5 panelist regular presentations, as well as 1 panelist and national presentation in this session. And before the start of this session, I would like to remind all our speakers, this session is being recorded. And we invite all the EU participants to submit your questions in English via the QA button. And you can find this button at the bottom of your screen. We will address your questions as much as possible after the talk or in the QA session. You will only see each question once. It is answered either verbally or in writing, when they will be displayed for everyone to see. You have the option of switching on the subtitles. As we have a huge audience, if we don't have time to answer all the questions we reserve over the next 90 minutes, we will aim to collect written responses from presenters to place on the conference website following this event. Please note that the chat box is disabled during this session. However, we ask you to keep an eye out for any useful messages that may be shared in this box from the organizers and moderators. We also want to remind you that you can see an excellent selection of submitted conference posters online at www.IWRA Conference.org. These posters can be found under the posters menu. And if you have any questions on these posters, you can send us an email to online conference@iwra.org, and we will contact the poster author. If you experience any technical problems, please Google Zoom Help Center or send an email to online.conference@iwra.org for assistance. Now, I would like to invite our keynote speaker, Professor Damia Barcelo Culleres from the Institute of Environmental Assessment and Water Research, High Council for Scientific Research of Spain. The title of the presentation is Fate, Risk, and Remediation of Emerging Contaminants, Antibiotic Resistance Genes, and Microplastics in Surface Waters and Groundwaters at Global Scale, Challenges and Solutions. Let's welcome Professor Damia to give the presentation.
Thank you very much, Xinghui Xia. Thanks also to Gisela and the people who invite me. By the way, I have been in Beijing Normal University many times before the pandemic, so next time I visit you. So I was asked to give a presentation of 15 minutes, so I will go as much as possible fast and give you an idea about what is it. Can I get the next slide, please? Emerging contaminants, you know, is a hot topic or threats that people is worried. What we understand by emerging contaminants, we understand pharmaceuticals, pesticides, personal care products, disinfection byproducts. This is a group of chemicals. Then lately, or in the last years, and it's still very, very hot topic, is antibiotic resistant genes, including as well other microbiological aspects like virus, and especially related to the COVID-19. Microplastics is a hot topic as well, and all these are cocktails which are in the water. I want to also give you some ideas about remediation, water treatment, and challenges and solutions, and an idea on the frame of this project, of this presentation. Can I have the next one, the next slide? I will show you some data, global data. This is, for instance, in projects in Europe that we were in charge, the Global Aqua projects, which are data from different, more than 300 contaminants, measures, pesticides, whatsoever in river basins in Europe. What we see from the hard risk, just to give you a brief outline, when you put everything together, but one thing is the concentration, the other thing is the risk. If we look at the risk, the yellow color is pesticides. So still pesticides are very risk to the environment and to the aquatic environment. And that's something that you need to keep in mind that when you look at all these chemicals. The next one is not from myself, it's from Alistair Boxall, a very well-known paper, who did the measurements around the globe on pharmaceuticals. And it's a very interesting topic because it has data from many, many rivers, and it was published in PNAS. And of course, the main pharmaceuticals, usually acetaminophen, metmorphine, carbamazepine, we know which ones are always found. The next one is also a global paper from Pedro Alvarez from the US, who also did the data from different places and also has got similar conclusions, also looking at the risk assessment of the different pharmaceuticals, what kind of treatment and things like that. So I think there are already, the pharmaceuticals is a hot issue at global scale. The next one is from my colleagues in China, Qian Sui, in Shanghai. I also collaborate with her. We look at the landfill. She is very worried about landfills, especially in Shanghai, and also pharmaceuticals in landfills. It's a hot topic and also something that needs to be considered because, there are many landfills around and of course, they contain also pharmaceutical. When we go to all these topics, the question here is wastewater. The next one is a recent paper that we did publish recently. The next one is about the microgenomic composition of the wastewater treatment. It's very complicated. What I want to show you with this slide, that the different treatments, they give different compositions of the genomic of the different taxa. And then what we know here is from this paper in Chemical Engineering Journal is that the taxa can be grouped according to the water treatment, and these are in more than five treatment plants in our region. So it is important to look not only to the chemistry or to the genomic composition of the water treatment to better understand the processes of degradation. Let's go to another topic or emerging topic. The next one is about antibiotic-resistant genes. It's from my colleague Barbara Capricci from the UK. She did in the area of Bristol, and she found here the One Health concept, as you know. So looking at antibiotic-resistant genes. And here what we can see is that, for instance, antibiotic-resistant genes is a hot topic, especially levels are higher according to the cities, especially cities, or also, of course, what is related to the farms and things like that. So antibiotic-resistant genes is a very, very important topic around the globe, and is increasing the number of people who are being affected, even death, people who are dying about this topic. The next one is related to something that we did as well, antibiotic-resistant genes during the pandemic with my colleagues in India, Manish Kumar. And in this particular case, the hot spots, just to give you the idea, is that the antibiotic-resistant genes or the resistance was related to the places where it was more COVID. They were looking COVID in the wastewater, wastewater-based epidemiology. And in that particular case, because they were giving to a lot of administration, and the administration was related to the use of a lot of antibiotics, the antibiotic-resistant genes were high levels in the same places where it was a lot of COVID, a lot of SARS, CO2, COV2. This was in Ahmedabad, which is around 7 million people. In the next slide, I want to show you another topic, which I did together with some colleagues in China, in Hong Kong, is the use of disinfection byproducts. Disinfection byproducts, is a very old stuff, trialomethanes and other ones. It's well known that they are formed in the drinking water when you chlorinate. But what they noticed, because during the COVID, they was using a lot of chlorine and disinfection products, they found higher levels in the Hong Kong area related to the use of chlorinated compounds. So the COVID was good. That will be my message at the end. The COVID was good for people, but not for the environment, as you will see later also for the other examples related to plastics. Next one is microplastics. Microplastics is a hot topic, as you know, everywhere, increasing reducing the number of microplastics jumping into the rivers. Recently, we did, 2 years ago, a study in Europe about how much plastic is going to the rivers, the RIMEL project, coordinated by the European Commission. In this particular case, we were evaluating the number of tons, around 5,000 tons per year in this project. By the way, the modeling papers which are related to the use of microplastic around the globe, they always overestimate the number. They always give you 2 or 3 times higher. Measurement microplastics in rivers still is a very important topic because many papers up till now are related to just to the modeling, and it's very important to measure. But people is now worried about microplastic, but my last comment here in this slide is that microplastic was the first paper on microplastics in the ocean, was published in 1972, 50 years ago. So we think sometimes that topics are new. They are not new anymore, but it was sleeping, this topic, but just lately it came to the surface. So can I have the next one? The next one are the main rivers in Europe, which are of course Douro, Guadiana, and Rhone, which are the different colors indicates the floating macrolator. This was the collaborative project in Europe, which I recommend you to read because it contains a lot of information related to that. In the next one, with some colleagues in Italy, they are proposing, they are working on biology, they are proposing the mussel watch program for microplastics, using mussels as a passive sampler for microplastics. Now I am getting close to the end. So I think this is also important now because people is worried about microplastics. If mussels have been used in the past for persistent pollutants, why not to use them for microplastics? Can I have the next one? And then when we go to the treatment solutions, there are many treatment solutions. I just want to give you some here list of techniques. Everything can be solved. This is I am very optimistic, but you need technology, membrane technologies, more friendly technologies, eco-friendly. Algae technology have been in the last years have been very much also in place because has also other effects like capture of CO2. You can remove with different oxidation techniques and other many different methods. So I think it's possible to solve the problem, but of course you need treatment. It will be more expensive. So can I have the next one? An example is here. In the next one is the example of the tertiary treatment, eliminating of antibiotic resistant genes. This is in a plant in Spain. We did with some engineers in Spain. So advanced oxidation process could eliminate the antibiotic resistant genes, but of course you need technology and you need more money. I just go into the end on my last slide, which summarizes all these topics. So what we had is just be aware that now we are after COVID. We are still some places COVID, but the world is open again. COVID has brought us a lot of pollution, and I think now we need to reconsider all the monitoring programs that we did in the past because the levels will be higher. We have higher levels of antibiotic resistance, pharmaceuticals. There are many examples now in the literature. Antibiotic resistant genes, disinfection byproducts. So there are many examples. So we need to be careful with this. So the single-use plastics still are in place. Still the number of tons used. Bioplastics are still very, very small amount. We had hygienic theater. Even the PCR diagnostic test, they contain up to 37 grams of plastic. So can you imagine how many diagnostic tests have been around the world? So I think we need to, of course, now there are technologies we can remove this. We can also remove the antibiotic resistant genes, and we can remove the SARS as well. We did some work together with some colleagues on the SARS removal. We need more bioplastics. We need, of course, less or more friendly chemicals, and we need less chemicals. And the last comment that Gisela would agree because she's also in the same line, we need to consider not just one chemical, the cocktails of chemicals, nanomaterials, we have antibiotic resistant genes, all these pharmaceuticals, pathogens, pesticides. So sometimes researchers, they only look to one group of chemicals, and it's a mistake because what we have in reality is a mixture, which is very difficult to evaluate from the toxicity point of view and from the effects on the environment. So I hope I did my time because I still have some, I see that in the screen I still have some time according to this. So I think we need to consider all these chemicals, and this is very important for all the studies going into this direction to look at the cocktails of chemicals and to look at how can we solve this, and this can be solved, as I told you, by good technology. So I think this is it. I don't know if I still have these 4 minutes, but I'm done. If you want to make some questions or later on, I can take it. So thank you. Sorry for being a bit fast, but people can use my presentation for anything if you need, and if you have questions as well.
Thank you, Professor Daniel. Very excellent presentation. And Gisela, would you please read questions from the Q&A box? So we have many time for discussion.
So
Yeah.
thank you very much for this, for your presentation. It's always lovely to see how you can address a lot in a few minutes. So thank you. And I would like you to discuss a little bit with us, because when I see all that, and then I think about Brazil and of course all the countries that we are struggling with, hungry and all very acute issues. How do you think we should proceed? Because sometimes we are doing this research that is so sophisticated and also trying to convince people, but it's hard because this is something that can be viewed as a minor problem compared to the others. So I would like to hear your opinion. What do you think about how should we do the outreach? Not to make people crazy about it, but to make maybe people think about that this should be included in their agenda.
No, that's a good point. I've been in Brazil many times myself, so I know the situation in all countries. So I think we should offer not the we need cheap technologies. I always say we need cheap methods. We need things that can be done easily and bioassays. So and is the way, if we go to the authorities saying this will cost you millions, they will say no. We should go for reasonable approaches, which are maybe less like, we cannot compare our countries with Switzerland or very wealthy countries. They can spend a lot of money. Even Spain, we are in between. So I think we should go for reasonable solutions, and then at least to do something. At least if we have a treatment plan, that the treatment plan works, this secondary treatment, because I have seen in Brazil, by the way, because I was there many times, as you know, many plants that were only just the blocks there, but not really working. So at least we should have operational systems working at the if you want at basic level, and try to say to the authorities, if we get a better environment, we also have better health for the people, because at the end of the day, if we don't have a clean river, or we don't have a good quality of the ecosystem, people will not be happy. People can get poisoned, and the environment, the biodiversity loss, all these kind of things. So, but we need to go step by step. I know the situation, because always environment, for many politicians, it costs money. It's not something that we can say, okay, this is cheap. No, it will cost you something, but you should also consider that if you have, I always try to sell this idea that if you have a very clean environment, a very clean river, you can also make money. You can have, restaurants, you can have live swimming, you can have boating. So you can use the ecosystem services concept, We should go in this direction in order to help politicians saying, okay, if we clean the river, it's not only the environment, it's also society benefits. I think we should go in this direction in order to help the society, and also the same with the parks, clean parks and things like that, going and cleaning, removing the plastic from the beaches. This will help also to have better people tourists going to the beach, and really not being afraid of getting any disease. So I think this is approach, but I know it's difficult, it's not easy, and I always say we should have technologies which are very sophisticated, but we should also have very cheap technologies. For instance, I don't know, to remove arsenic in India, which is a very well-known topic for many years, and we should have simple technologies, and also bioassays, very very easy ones to that can be performed in any place. So this. Thank you.
Thank you very much. So we have a question here from Sabri Hanna. Thanks. Any studies about the effect of microplastics on ecosystem and its accumulation in organisms like algae? And what about its treatability?
There are many now. There is a boom of papers on microplastics in the literature. So there are plenty of them on algae. I don't know if there is many papers. Not many. Most of them are on fish, but the treatability of when you have the plastics can be treated in the water, when you have it in the organism, it's not possible to treat the organism. I mean, but when you are in the water, yes, there are many methods now, even advanced oxidation. There are different technologies that you can remove it, but you should do it in the treatment plants before going to the receiving water. No, so that's the question. And of course, we should use less plastic, but the message should go to use less plastic and more biodegradable. But at this moment, biodegradable plastic, I think, is only 5–10% in the world. So it's not much because it's more expensive. And it's always the same thing, you know. The manufacturing people or the people who wants to sell, they want to use something which is very cheap, and everybody can use it and buy it. No, I mean, but we should go more in the direction of bioplastics and degradable plastics.
Okay, thank you. Thank you,
Okay.
Professor Damia. So we would like to move to our next presentation. It's from Gabriely Groto Militão from the School of Technology, State University of Campinas. The title of the presentation is Mutagenicity Evaluation of Rubber tile oxidant byproduct 6-ppd quinone using the Ames assay. Now let's welcome Gabriely to give the presentation.
Okay, so hello everyone. So I'm Gabriely and I'm a master's student at UNICAMP, Brazil. I'm here to talk about our work, mutagenicity evaluation of the rubber tire oxidant byproduct 6-ppd quinone using the Ames assay. Can you pass the next slide, please? So first of all, I have to talk about the 6-ppd quinone sample, which is a transformation product of the rubber tire antioxidant 6-ppd. And this compound reaches the aquatic environment by stormwater and urban runoff. Can you pass, please? So there is a limited information about the toxicity of 6-ppd quinone, but it's already considered a relevant emergent contaminant because low concentrations are correlated with fish mortality in urban areas, especially true in salmon. And some laboratory studies shows that 6-ppd quinone doesn't cause actual toxicity to some aquatic species like Daphne magna. And so the objective of this study was to verify its mutagenicity using the Salmonella microsome assay. Can you pass, please? So for the microplate agar test, we performed a miniaturizing version of the Ames test according to ZARG. So the first step was to add 2 microliters of the sample plus 50 microliters of the cell suspension in one tub. Then we transferred 26 microliters to another tub. In one tub, we added 25 microliters of phosphate buffer, and in another tub, we added 25 microliters of S90 mix. So we pre-incubated those tubs at 37 Celsius degrees for 90 minutes with shaking at 180 RPM, and after that we added 1 milliliter of molten agar per tub and distribute 250 microliters in 4 wells. So plates were incubated after that at 37 Celsius degrees for 66 hours, and then colonies were counted. Can you pass, please? So the sample 6-ppd quinone was from Toronto Research Chemicals in Canada with a purity bigger or equal 97%, and we dissolved this sample in DMSO at the limit of the solubility, which was 5,000 nanograms per microliter. DMSO also was used as negative control, and we tested in concentration response with a maximum concentration 100 nanograms per microliter. We selected 5 strains to the test, so YG1041, TA100, TA98, TA102, and TA5035 to test with and without metabolic activation with 5% red liver. To TA100, we performed an additional test with 10% of S9 mix. Can you pass, please? So our results present no statistical increase in the numbers of revertants per well for YG1041, TA98, TA102, and TA1535 in the presence and absence of metabolic activation. Also, no statistical increase for TA100 in the absence of S9 in any concentration tested, but shows the difference in the presence of S9. Also, no signs of toxicity was observed in the background of the agar plates. Can you pass, please? So here we have the statistical analysis with TA100 in the presence and the absence. Here is only in the presence of S9, which performed using an ANOVA with a linear regression model, Bernstein. And we can see only a weak increase in the number of revertants per well was observed inserted from 6.25 nanograms per microliter in 2 independent experiments performed 5% S9 was the test A and B, and increasing the percentage of the S9 to 10% since to turn the compound less active because mutagenicity started from 12.5 nanograms per microliter. You can pass, please. So we conclude that 6-ppd quinone showed a weakly mutagenic response with TA100, which caused base pair substitution only in the presence of metabolic activation, S9 mix, red liver, and an increase in, like I said before, an increase in the number of revertants per well was observed. It started from 6.25 nanograms per microliters in 2 independent experiments performed with 5%, and with 10% doesn't seem cause a higher increase because it started 12.5%. So more tests will be conducted using less concentrations in start solutions to verify if the lack of mutagenicity in the higher concentrations is due to the compound agglomeration in the incubation mixer, and also mutagenicity in vival tests are being conducted using aquatic invertebrates to confirm the in vitro findings. I just want to add some information here. Last week was published a paper that shows 6-ppd quinone is capable of causing DNA adducts in mammal cells. So these results confirm our results, and you can pass, please. I want to thank you, the University of Saskatchewan, especially Dr. Brickman, who provided the sample, my university, UNICAMP, my lab, and my professor, Dr. Gisela, who is here, and FAPESP for research and funding. Thank you for the opportunity to be here showing our work, and thank you all for listening.
Thank you, Gabriely, for your excellent presentation. So I would like to move to our next presentation. The next speaker is Natalia Soares, coordinator from Florida International University of United States. The title of the presentation is Assessing Forever Chemicals in South Florida Aquatic Environments and Potential Environmental and Human Health Risks. Let's welcome, yeah.
Hi. Okay, yes, I can start. So yes, thank you so much for introduction. Good morning, good afternoon, depending where you are. We can go to the next slide. I mean, I'm an assistant professor at FIU, and I'm going to be talking about PFAS. So just a little bit introduction, since I do want to show like I have some interesting results. So I practice to be able to be finished at 10 minutes. So PFAS stand for per and polyfluoroalkyl substances. They have been synthesized for such a long time, like 6 years ago. They're very stable. And this has actually led to their, because it's presence in the environment and accumulation in humans. They are different from the traditional pollutants. So they have hydrophobic and hydrophilic properties. So they tend actually to bind preferentially to proteins. And, I'm showing here, two of the most common molecules that have been present in the environment, like PFOS and PFOA, even though they have been banned. The problem is, most of the alternative compounds, you can show the next slide. They're still, as you can see, yeah, it was the last one, sorry. They're still, as you can see here, they're still perfluorinated. So the example here from Gen X. Okay, next one. So as you can see here in this figure, these substances are present in many consumer and industrial products. Okay, I'm not going to be talking specifically each of them because I don't really have that much time. So they are actually present, they have been using for different applications, and they're known to have a wide range of diverse health effects in humans and animals, even potentially carcinogenic. Next one. So I just wanted to show with this slide, how they can be transporting it and how they can actually end up in groundwater. We can see here the input from industrial use, from PFAS, wastewater treatment plants, landfills, crops and farms. So they all can actually run off or leach. The leachate can actually go into nearby water bodies and then eventually infiltrate it to groundwater. And you can even have the input for non-point sources, like for example, also some rainwater. So the EPA Health Advisory Guideline, was set up in 2016, which included only PFOS and PFOA. That the level in drinking water can also surpass 70 nanograms per liter. More recently, this guideline has been updated, and you can see that the levels are a lot lower now and has included some of the other PFAS, PFBS, and Gen X. Next. So the objective of this work is to try to see what are the currents, levels, composition, and distribution, and try to identify some of the sources here in South Florida. We still, we need also more information about those emerging, alternative short-chain PFAS, and understand, how those contaminants can be contributing to both, aquatic and human health risks. So this picture here is from a review paper that we wrote a while back, but recently 2 years ago, that just you can see there's still a lack of studies in Florida. And, one of the worries here in South Florida, you can see that the levels has been found to be high in groundwater. Next. So we have, just to show that we have been doing some comprehensive sampling. We have collected surface water, tap water, and as well, we have collected rainwater all over Florida. And we also going to be presenting today some results from oysters that we have collected also from here, similar locations. Next. So this is, we have developed a methodology for, that is very in line with the EPA methods for measuring PFAS in water samples. So we have, we take about 250 milliliters of water samples, amended with internal standard, and then it goes to solid phase extraction. So we have a semi-automated system here, has been completely modified and using weak anionic cartridge, exchange cartridge, and then measurement by LC-MS. And this was being developed by one of my graduate students that just graduated recently. Next. Oh, and then, this was developed by the postdoc in our group, extraction method for biota sample for the oysters, where they were homogenized using an ultra-Turux probe, using basic methanol, and then were cleaned up with fluorosil and graphitized carbon black before LC-MS analysis. Next. So this is, okay, finally we are in our results. Here we can see PFAS distribution that we found in tap water, and it kinds of in the three locations, the east, west, and central Florida. We can see some of the major PFAS, some shorter chain like PFBA. Also, we still see a higher level of PFOS and PFPEA. Next. And comparing these three locations, what we see is that you see the highest concentration have been found to be the east coast of Florida, followed by west coast, and then central Florida. Next. Looking at these concentrations of PFAS in surface water here in Florida, we saw also similar profile. We saw like a highest concentration was in Biscayne Bay, followed then by Tampa Bay. I mean, let's ignore for now this highest level here, this highest bar here in red, and then following by some of the canals and that leads to come from the Everglades, and then the lowest concentration was in Key West. So this the story about this canal, as you can see, we sampled them like coming all over. You can see in the map from the Everglades, and then as it passed by here from Homestead Military Air Base, that's where this highest concentration of total PFAS you're seeing here. So one of the predominant PFAS found, it was PFOS, with concentration up to 48 nanograms per liter. I wanted to point it out, like, you know, Florida DEP, they regulate in saltwater these levels for 13 ppb, so they're a lot higher than what we're finding it. But if you go to, you know, more restricted guidelines, such as in Europe, Australia, and New Zealand, you see that if you actually wanted to, for the purpose of protecting it, this aquatic life, you know, the levels that we have found here, you know, could actually be potentially affecting it, marine life. Next. So here, you know, just showing very shortly some concentration from rainwater. We can see that the highest concentration we saw for total PFAS was 24 nanograms per liter, and we look at PFOS and PFOA. We see that they were actually, you know, even higher than the guidelines, you know, they're coming from, you know, from the rain. And we see also some very interesting profile in the differences from rainy season and dry season. With the rain season, you see having a higher amount of short chain and the dry season long chain. Next. So what about the oysters? We also look at, you see, we found like PFAS all over here in the oyster samples with PFBA, PFOS, and PFTEA having like the highest concentration. Next. We found also that Biscayne Bay was the one that has shown a significant higher concentration compared to the other two locations, Tampa Bay and Marco Island. We also found out that, you know, the sum of the PFAS was negative correlated with some index coefficient index, shell thickness, total weight, and animal weight. So next. We also did calculated some risk assessment for human, you know, based on drinking water consumption as well as oysters. As you can see here, we calculated the estimated daily intake from individual PFAS as well as the hazard index. When you consider here median exposure, we see that the hazard index was a low risk, but, you know, with the high exposure, we are actually seeing a really higher risk of exposure to those contaminants. But for oyster, we actually found the hazard index was of a low risk, so below 1. Next. So just one, you know, some takeaway message. The Florida, we have seen that, you know, Florida coasts have been exposed. We found PFAS in different types of medias, with Biscayne Bay being one of the most impacted areas. We have found most of the concentration that were identified, you know, the highest one identified in areas like close to airport, military air base, you know, and, you know, some of the Biscayne Bay canals. We have also identified some of the most predominant PFAS that were found in tap water, surface water, and oysters. And that strong negative correlation that we found between total PFAS and weight and shell thickness, you know, that kind of indicated that we might have an oyster development impairment. And what I wanted to say is that the risk assessment, even though we found some potential health risk to, you know, it has to be careful interpreted because of the available, you have a lack of available reference doses in the literature. Next one. In 7 seconds, I just wanted to thank you, all my group, postdoc, graduate student, and undergrad. They have done like an amazing work, the Institute of Environment, as well as funding from NSF Crest. Thank you.
Thank you, Natalia, for your excellent presentation. Yeah, very interesting research. Yeah, so we would like to move to our next presentation. It's from Raymond Dominguez. from the State University of Campinas, Brazil. The title of the presentation is the Occurrence of Emerging Contaminants in Pampulha Lake, anthropic pollution of a UNESCO Heritage Site. So, Raymond?
Hello, I'm Ramon, and this is my presentation on the occurrence of emerging contaminants in the Pampulha Lake in Brazil. Next slide, please. The Pampulha Modern Ensemble is a visionary city garden project established in Belo Horizonte, capital city of Minas Gerais state in Brazil. It was built in the 1940s and includes a ballroom, a museum, a sports club, and the famous San Francisco Justice Church. The project was declared a UNESCO Heritage Site in 2016 due to its importance to Brazilian modern architectural identity. Next, please. All the buildings were designed around the Pampulha Lake. The Pampulha Lake was initially thought as a possible water source for the city, but since it's located in a very urban area, it was very damaged by pollution, and its water is no longer suitable for human consumption. Next. Considering that, the present study aims to identify the occurrence of many emerging contaminants in the Pampulha Lake. The contaminants analyzed are classified here as pesticides, pharmaceutical and personal care products, or hormones. Next. For that, 6 sampling points were established in different areas of the lake, and 2 sampling campaigns were carried out so far, both of them in dry weather. All the samples were extracted through solid phase extraction, and they analyzed, quantified by liquid chromatography coupled with mass spectrometry. Next. So for the preliminary results of this study, 7 of the compounds were not found in any of the samples so far, which is especially interesting for caffeine. Caffeine is a well-known marker for pollution by untreated sewage. The Pampulha Lake is reported to receive untreated sewage from its surrounding neighborhood, but this may indicate that that doesn't end up being a major source of pollutants to the lake. Beyond that, the major contaminants found were atrazine, simazine, carbendazine, and 2,4-D, all of them being pesticides widely used in Brazil. Next. Here's the list of contaminants that were found at lower concentrations on levels below 50 nanograms per liter. Most of them are classified as pesticides. Next. And this is the list for contaminants found on concentrations ranging from 50 to 600 nanograms per liter. Right here, if you take a closer look on simazine, for example, that's presented in gray, it's possible to observe that there's a higher concentration of this contaminant in sampling points 1 and 2 when compared to the other 4. This may indicate a possible distribution pattern for this contaminant, and it's observed for many of the analytes comprehended in this study. And next. So considering these distribution patterns for simazine, for example, that presented a higher concentration on sampling points 1 and 2, that's the region of the lake that is closest to the buildings and the gardens from the Pampulha Modern Ensemble. This may indicate that these pesticides may be used in the maintenance of these gardens and enriching the lake water as a result of this application. Where fipronil, on the other hand, presented a higher concentration in sampling points 5 and 6, that's the region of the lake that receives the largest numbers of tributaries, indicating that maybe these contaminants are carried out through the tributaries and reach the Pampulha Lake as its final destination. Next. So we expect that with the future sampling campaigns now performed during a wet weather to assess the seasonal effects on the level of these contaminants in the lake, and also to confirm the distribution patterns observed so far for many of the contaminants, as a way of discussing the possible sources of these contaminants to the lake, and with that, discussing the more appropriate measures to prevent the pollution and improve the water quality from the Pampulha Lake. So, next. I think that was it, a very short presentation on its occurrence, and feel free to contact me if you're interested. Thank you for the opportunity.
Okay, thank you, Raymond. Yeah, excellent presentation. Yeah. Now let's welcome to our next speaker. Yeah, Nicolo Brankovic from the Soil Physicals and Land Management Group, Wageningen University of Netherlands. The presentation title is Transport of Veterinary Pharmaceuticals in Lowland Catchments, a lumped modeling approach. Now let's welcome Nicolo.
Okay.
Yeah.
Hello, everyone. I'm Nicola, as introduced, and today I will talk about the transport of veterinary pharmaceuticals in lowland catchments and our modeling approach. Just to highlight that this work has been done as a cooperation between Wageningen University and the EPFL in Switzerland. Next slide, please. So veterinary pharmaceuticals are compounds that are typically used to treat or prevent animal illness. And usually they are characterized by quite diffuse and complex hydrogeochemical dynamics. They might have negative environmental effects, and this is one of the reasons why they are also highlighted as contaminants of emerging concern under the group of pharmaceuticals. And as we have seen from some of the previous presentations also, that so far environmental monitoring and modeling focus has been mostly on human pharmaceuticals, while there is still some gap and some lack of knowledge in these kind of things for veterinary pharmaceuticals. And if we go into more details, their transport towards groundwater and possible soil plant contamination has been also partly addressed over the past decades, while the transport of these kind of compounds to and along the stream network has received much less attention. For this reason, it has also been highlighted by the European Union strategic approach to pharmaceuticals as one of the relevant research questions. Next slide, please. So what we have done so far and what we are working on at the moment is the transport of VPs in the environment. And in the first rectangle, we call it phase 1, you can see that their introduction in the environment starts from the usage. So these compounds are being given to the animals, and then a portion of these compounds goes out of the animal's body via excretion of manure and urine. And typically in the Netherlands, maybe this is not the case in other countries, but in the Netherlands, this manure is stored for a certain period of months, around 6 months, which allows for these compounds to be partly degraded. For some of them, the residues still end up in manure which is applied on the soil, spread on the soil. And from that point, they can further transport, if you look into the rectangle 2A, either to groundwater, or they can further transport to surface water. Transport to groundwater, I will not address much today. We will focus mostly on the VPs in surface water. So next slide, please. Very briefly about steps 1 and 2. So application to soil and the leaching to groundwater. We have developed a national scale model for the Netherlands, where basically for each of the 1.2 million fields, we estimate the applied amounts of pharmaceuticals to soil, and we also estimated the leach quantities to groundwater. As you can see, we have a national scale model, but we also have the option, we have the data to estimations, let's say, for the local scale. What is a bit problematic for the local scale is that you cannot, let's say, freely work with these kind of things, because if you want to publish some results or similar, you might have problems, especially now with the nitrogen crisis, where none of the farmers, which is reasonable, doesn't want their field to be pointed out, let's say, as a problematic in sort of contamination or similar. So far we are keeping these kind of things on a local scale just for research purpose, and we are mostly focusing on a national scale model, and we have published these results in 2 scientific papers. Next slide, please. So what is the main topic of today's presentation is transport, as I said, to and along the stream network. And this route is quite indirect and usually very difficult to quantify. And what is important is that the pollution of surface water can occur much downstream than the application areas, and then to trace the exact source is quite difficult. And the degree of this contamination depends on many processes, like the way manure is applied, the physical characteristics, distance to surface water, etc. And since we're working for the Netherlands, where you have typically lowland catchments, all these processes and their interactions are becoming even more relevant because you don't have the slope-driven transport. And so then it's very important to address these processes and to know which one contributes into which degree. Next slide, please. So what are the main challenges in research questions? So how to translate the data and the estimations from field scale to catchment scale, where you have a lot of things mixing together, both surface and subsurface contributions. Then how are these compounds transported to river network, how quickly this transport occurs, how much are they degraded along the way, and can we validate our predictions? I'll try to answer these questions now. Please, next slide. So what we did, we selected a typical agricultural catchment in the Netherlands with quite intensive livestock sector. And together with regional water authorities, we selected some VPs that are known to be used in that region. And then we performed some estimations for the concentrations that are applied on the soil and for the manure application patterns, characteristics of these compounds, and some local meteorological conditions. And we use this type of data as an input into, please, next slide, into an existing rainfall runoff model that's also developed at Wageningen University, and it's called Walrus. This model basically predicts relevant water fluxes, and I will not go into details with this kind of modeling, but just to highlight that we basically distinguish three important reservoirs, three important routes, and that's through the soil, and that's through the quick flow, let's say runoff, and then what ends up eventually into the surface water. With this model, we estimate water fluxes, and then we with some numerical schematization and then our input data and calibration, we add also the component of the veterinary pharmaceutical transport to this. And eventually out as an output of this model, we get the concentration of veterinary pharmaceuticals in the surface water. Next slide, please. And then what are we doing actually to validate our predicted concentrations in the surface water? So as you can see, this is the same area that was selected earlier. We picked up 4 monitoring locations, again, selected with the local water authorities, and we installed passive samplers there. As you can see on the right picture, these types of samplers are called speed discs, and they have kind of adsorption material to which veterinary pharmaceuticals attach. But for this, you need to have a certain quantity of water passing through them, and that's why it was very important to pick up the locations that are meaningful, and that's why we also did this with local water authorities. And then we targeted 46 VPs in total, and some of them we will use for validating our concentrations. What is also interesting is that we don't have exact quantification yet, but we found some disinfectants in the area, and this is, for example, quantified for the first time in the Netherlands. Most probably these type of compounds come from the cleaning stables and some animal equipment disinfection set, etc. Next slide, please. So just to summarize, we illustrate a modeling approach to investigate the temporal dynamics of veterinary pharmaceuticals in lowland catchments, as typical in the Netherlands. And we combine an existing rainfall runoff model with estimations on soil applied veterinary pharmaceuticals and some other parameters to build a veterinary pharmaceutical transport model. Our results also provide some insight into the dominant transport routes, basically, if you remember those 3 reservoirs. So by quantifying the mass that's transported with them, you can see which of the routes is the most dominant, in which period, in which season, and you can focus maybe more or less on certain components of the transport. And since we also address with our model the residence times in surface water, we are trying to better understand the interaction between the agricultural activities like manure application and water quality. By getting an insight into this, we hopefully will be able to better identify suspected risks and eventually suggest some regional or local policy improvements. And I think that's all. Thank you. Right on time. Thank you very much.
Thank you, Nicole. Yeah. Now, we would like to move to our next presentation. It's from Marisa Fernandes from Federal University of São Carlos, Brazil. The presentation title is Bioremediation of Azodine Tartrazine Yellow by 3 different microalgae genera. So Marisa, would you please?
Hi, thank you. So, hello, my name is Marisa. I am a master's student from Federal University of São Carlos, Brazil, and my work is about the bioremediation of azodine tartrazine yellow by 3 different microalgae genera. Can you go, please, the next? Okay, the objectives of my study were analyzing the growth and degradation capacity of 3 axenic microalgae species in the presence of tartrazine. We used the green microalgae Senescens spinosus, one not identified Chlamydomonadalis, and Muriella decolor. The strains were obtained from the freshwater microalgae culture collection of Federal University of São Carlos. Next, please. Thank you. So, tartrazine is a dye widely used in the food industry. It's a nasal compound with orange-yellow color and can affect human health, in addition to play a negative role in aquatic communities. In the pictures, you can see some examples of tartrazine food and the effects in our health and environment, like allergies and child hyperactivity, and in the environment, low dissolved oxygen concentration and eutrophication. You can go, please. Okay. Now, in the slide number 4 is about the methods of my work. Each microalgae was cultivated in triplicate under 3 conditions. Conditions 1, it's a control where we had the only microalgae in culture medium. Condition 2, a light quality control where we placed a flask with tartrazine solution between microalgae culture flask in the light source. I call it the asset control, as I showed in the previous slide. And condition 3, that is the treatment with the microalgae in middle with tartrazine. Can you press enter, please? Again. And we also performed a control for abiotic degradation of tartrazine in culture medium without algae. In the next one, please. Okay. So in the next slide, regarding the dye degradation, the not identified Chlamydomonadalis and the Muriella decolor could significantly degrade tartrazine compared to the treatment of abiotic degradation of tartrazine. They degraded respectively 24.7% and 21% of the dye. And although we detected dye degradation by Senescens spinosus, it was not significantly different from abiotic degradation. Next, please. Finally, in the slide number 6, the comparison of the microalgae growth curves between treatment of Muriella decolor and Senescens spinosus shows that the presence of the dye was neither toxic nor affected the growth of the algae due to the change in the light quality, since there was no significant difference in their growth between the treatments and the controls. Just to remind you, the SEP control is the flask with tartrazine solution between microalgae culture flask and the light source. And regarding the not identified Chlamydomonadalis, the cells formed a biofilm on the flask wall, which hampered the analysis of growth. On the other hand, these results indicate the possibility to study this strain to be used in biofilms for effluent treatment. And just for the record, before we performed the main experiment in flask, we tested this strain in tubes, and the results showed no difference between treatment and control. And finally, the relevance of this work is because it's an initial step towards bioremediation of disease. There is a little discussion about the tartrazine biodegradation in literature, and the future research may help of its removal from effluents of the food and textile industries and their proper disposal in water bodies, contributing to a healthy and balanced ecosystem. That's it. Thank you very much for your attention.
Okay, thank you. Thank you, Marissa. Yeah.
Thank you.
Yeah, let's move to our the last presentation is from Edson de Souza Ramos from University of Brasilia of Brazil. The title of the presentation is What Contamination in a Multi-Industrial Region in Brazil, Decision Support for Water Quality Evaluation and the Management of Contaminants of Emerging Concern.
So, thank you so much. My name is Hadassah, and I am a postdoctoral degree associated with the AQUA group from the University of Brasilia. Well, I would like to begin. Please press enter. Please enter. Oh, oh my God. Okay. I guess we are all benefited by industrial activities whose products are of great importance in our routine. However, so many benefits also come at a cost, since industrial facilities pose a risk to the environment due to the production of large amounts of waste containing various contaminants. In particular, in developing countries, despite economic growth, rapid and severe damage to the environment can be observed, including biodiversity loss. And such damages, they are poorly investigated. Among the substances released in the environment are the so-called contaminants of emerging concern, such as pharmaceuticals, which I will focus from now on. And these kinds of contaminants are even less investigated in these countries. Next, please. Oh, yeah, some studies have estimated that industrial installations can release more than 100 kilograms of drug residue in aquatic environments. Meanwhile, a municipal station releases on average 23. Well, can aquatic life be in fact preserved? Well, let's now take a look at Brazil. This emerging country is an exponent in the production of generic drugs and medications in Latin America due to, among other reasons, the presence of this agro-industrial district of Anápolis, located in the Midwest region in the Goiás state. The Damia district is the second largest pharmaceutical pole in Latin America, and this district has only one wastewater treatment plant responsible for treating both sanitary and industrial wastewater, and it is equipped to carry out only primary and partially secondary treatment so that the final effluent is only partially treated. Unfortunately, this treatment station is undersized, and local reports state several non-compliance situations regarding effluent guidelines and recipient water body guidelines. Considering the importance of DIA in the drug production scenario, we set out to investigate the occurrence of emerging contaminants in water bodies impaired by partially treated wastewater releases. However, which ones? Which contaminants? Next, please. One of the biggest challenges associated with the investigation of emerging contaminants in industrial effluents is the complexity of the waste, whose composition is basically unknown. In order to identify possible substances for investigation, we started with a non-target screening approach through high-resolution mass spectrometry and the use of mass spectra databases. And through this strategy, we were able to identify several drugs, including erlotinib and tropisetron, which are substances very they're very uncommon in Brazilian waters and has not been investigated until then. Then a method based on LC liquid chromatography tandem mass spectrometry was validated and was developed and validated to quantify emerging contaminants. And the method presented good performance. Now, where are we going to monitor? Next, please. Next, please. Thank you. Thank you. The discharge point, the DIA wastewater treatment plant discharge point, is located in a stream called Abraão. The stream flows into the Extrema River, and sampling points were selected before and after the confluence with the stream with the Extrema River, and sampling campaigns were conducted monthly. Several were next, please. Oh, sorry. Now here we can see a few photographs of these downstream sampling locations in which we can see a natural color, some foam, and also a strong odor similar to a rotten egg. Next, please. Several water quality standards were obtained. As we can see here, there is an increase in parameters values such as electric conductivity, phosphate, and chloride, whose occurrence in tropical waters is associated with pollution sources. We can also see a decrease of dissolved oxygen after the discharge point. And we can also see a gradual decrease as the distance decrease of these parameters as the distance from the discharge point increases, and poor quality can observed after the discharge point. Next, please. If we look at the concentration of emerging pollutants range, we can observe a very wide concentration range from low nanograms to micrograms per liter, over 1,000-fold difference. And to assess environmental risk, we use the risk quotient approach using restrictive NEC values obtained from the literature, which showed high risks for several substances, including endocrine disruptors, some antibiotics, and several anti-inflammatory drugs commonly used. Despite showing the potential risk, this approach only considers individual substances, so a broader approach is needed. Next, please. Well, for this broader approach, I'm going to use the Water Quality Index from the Canadian Council of Ministers for the Environment. I'm using this index because of its very flexible algorithm. This algorithm considers the number of parameters, number of tests, and magnitude of non-compliance with the guidelines used. It also allows you to choose the guidelines to assess whether there is a failure or not. This index was developed to report water quality of all sampling points in a single value, so it does not discriminate water quality per point. Next, please. So we are going to adapt this water quality index to report water quality per monitored point. And to do such, we excluded one of the variables that would become equal if we were to use them as the original algorithm. And to select which quality parameters best characterize the water body, we use principal component analysis. And as we can see now, the water quality index now tests for fault frequency and for fault magnitude. And the PCA was able to discriminate which variables are associated with higher and lower water quality. So we selected these 4 variables to adapt our new water quality index. For comparison purposes, we calculated the adaptive water quality using all the parameters obtained, and then after that, only the ones selected by the PCA. If the first case, all the parameters were to be used, water quality is not consistent with the number of failures. And second, it's way more consistent. So far, only physical chemical parameters were considered, but how about emerging contaminants? Next, please. Well, emerging contaminants, they are barely included in routine water quality analysis, and they do pose a huge threat to aquatic life. So our goal was to include these contaminants in the adapted water quality index, but our question was, which ones should we include? So we used as a guide candidates list from the APA, and also those which are widely investigated and pose a higher threat to the environment. So we chose 3 biocides, 3 endocrine disruptive compounds, and 3 anti-inflammatory drugs. And we decided to use as guidelines the literature values of NECs as guidelines. Next, please. And now, with the addition of contaminants of emerging concern in the adapted water quality index, we can observe that more restrictive quality index values were obtained, and water quality was poor to basically all points from the discharge point to downstream point number 3, which is over 30 miles away from the discharge point. And poor quality was observed for all points except for the upstream. And what is interesting to observe is that now the water quality index does not just consider the presence of contaminants of emerging concern, but also they are especially they are high concentrations that pose a threat to the environment. Next, please. So finally, the concentration of these contaminants from the contaminants of emerging concern were more than 10 times higher when compared to receiving impaired water bodies from municipal waste discharges. And this is the first time that contaminants of emerging concern were investigated in industrial sources in Brazil. And a simple and comprehensive estimate for water quality was proposed to guide decision making. I would like to thank all of you, to thank my university, my research group, and thank you for your attention.
Okay, thank you. Hadassah, yeah, thank you for your excellent presentation. Now,
Thank you.
let's move to our QA question section. Now, Gisela, would you please read the questions from the QA box?
Yes, hello.
Yeah.
I have a question for Hadassah from Fernando. In your opinion, what several impact in the healthy in the region's population?
I'm sorry, can you ask it again, please?
Yeah, he wants to know what are the impacts on the health of the region's populations.
Well, the region is basically used for the industrial purposes, and there are a few small farms around. Some farmers do wish to use the rivers and streams for drinking purposes to their animals, and unfortunately, these water bodies, they are so impaired by the industrial waste that they have visible presence of dyes, industrial dyes, so animals cannot consume these waters, nor people. Some local residents even report fish mortality. Whatever animals drink from these waters, they have acute diseases and just die. So the industrial toll does have a very negative impact in water quality and makes it unsuitable for animal consumption, for human consumption, and for aquatic life preservation.
Thank you very much. Now there is a question from Reina Sotilio. Congratulations to this event. And Ramon, please could you answer, have you considered cooperation among the countries on good practices and new methods and infrastructure to treat emerging pollutants, especially in dispersed populations that are faced with contaminants?
Yes, thank you so much for your question. As of right now, at the stage of the study, it's still very initial. We're still trying to get a better overview on its occurrence of the comprehended emerging contaminants in this study. With the results, better define the ways of which approaches we may take on improving the water quality of the Pampulha Lake. So as of right now, no solid actions have been taken for the treatment of this emerging pollutants in this case so far.
Okay, thank you. I have a question here from Molloy. I think it would be addressed to Damia Barcelo. Are you there, Damia? So the question is, thank you very much for your presentation. You mentioned that the available data on microplastic is from modeling studies, and we still need to measure in the environment. I agree with you. How far are we in terms of understanding the levels of microplastics in surface, marine, water environment? And can we conclusively say we understand the impact and interaction with environmental metrics so far?
Well, there is a big problem with microplastics that you are aware, I guess, is the analytics, the measurements. There is not yet a standard method on microplastics. Last year, it was an interlaboratory study published in Stotten, and it was a mess. It was a disaster. If you use pyrolysis GPMS or if you use FTIR, you cannot compare your data. So first thing that we need to do is to have good measurements. So for this reason, probably they were a lot of data on modeling, because, you know, you can always estimate how much plastic is being used and things like that. But we need to have better analytical methods which can be compared, because at this moment, if you find in Brazil, I don't know, 30 particles per liter, or I find 300, what does it mean? How did you sample? How did you measure? You know, we cannot compare it. So when you have a paper on microplastics, you need to look at the method, how this was done, and things, and every detail. So for me, this moment is the most relevant point. So comparison is still very difficult, and we need to find a way. There are people working on that, but the measurements and the standardization to use standard plastics for interlabs. So we need a lot on this and at global scale, not just in some areas. So I have been a lot in China before COVID, and China, they have very good infrastructure in some labs for this, but in Europe, we're still now getting more infrastructure, but not yet. In America, also less. So I think still a lot of work to be done. So you can do in your lab some studies, okay, but can you compare it with your neighbor? Is another issue, you know. This is my opinion, and I think I'm very critical with this, you know, because it's, of course, they're very interesting, the ecotox studies, and of course, at environmental level, if you do an exposure to microplastics or to any other pollutant, please use environmental levels, not use high levels, you know. Not higher levels, because otherwise it's not relevant. So we are still at the moment of measuring, which is still difficult thing, and many people are still using microscope, just simple microscope to show the surface. So it depends, what do you want to measure? Do we want to know the composition of the plastic? Do you want to know? So there is a very complex issue, and I think a lot of PhD students need to work on this, and a lot of groups step by step, you know. So I think I'm very critical with this. So it's not, when I see a number of microplastics, I'm always doubting about the number.
Okay, thank you. There is another question. I think it's for you. It's just a continuation of this problem. What are the main difficulties faced on the studies that use the methodology of monitoring microplastics in mussels? Then.
No, well, for mussels, it's not a problem. I mean, you can do an extraction, you can do the cleanup. I don't think it's a problem to collect the microplastics in mussels. Not more difficult than to do it. People think it's difficult. You know, mussels is a filtration element, organism, so it filtrates a lot. So it's and there are standard methods now for biological samples. So the question is, after that, when you have the sample cleaned, what technique are you using? How do you measure this? Do you want to know the composition? So how do you establish the method, and the composition of the plastic is easy, or I mean, it's easy if you have the right technique. But for the cleanup and sample prep, it's not a problem. I see more problems in the water, because, you know, in the water, different nets give you different results. If you use 80 micron nets or 200 microns, then you will have different types of plastics and amounts. In a bivalve, if you use a standard, mussels have been used in ecotoxicological studies during many years. It's very much a standardized method. So I think it's not a problem from the sampling point of view. And I think for me is the measurements and establishing the, let's say, the data and establishing what do you want to measure and what is the concentrations. Probably it will be better to have mussels around, standard mussels, of course, I don't know, Mytilus or anyone that is selected by you, by the ecotox people, and and then use the same muscles. Probably this will solve one of the problems of the sampling devices, because at this moment sampling is also a little bit chaotic around the world.
Okay, thank you, Damia. Yeah, Gisela, I have two questions. Can I ask two questions? Yeah, Gisela. Yeah.
Okay, we still have some in the QA. Should we continue with the QA, or you want to do your questions now?
Yeah, I want to ask a question for Nicola.
Okay.
Yeah, Nicola.
Yeah, so go ahead.
Are you here?
Yeah. Yes,
Yeah,
sure.
yeah.
Just a moment.
So when you model the veterinary pharmaceuticals in the catchment, did you consider the biodegradation of the pharmaceuticals, or just model the transport?
We consider also the degradation of pharmaceuticals. Yes.
You also consider biodegradation. How about the half-life of the pharmaceuticals in the catchment?
That's a very big problem, because you have a lot. We do not perform investigation by ourselves, but we use literature data, and that's quite different between the regions and countries, etc. So we are trying to use something that's available for the Netherlands, preferably from experiments performed somewhere nearby. But yeah, I'm not so confident in the values we are using always. So that's why we are also trying to calibrate some type of decay rate and degradation rates to compare with our predictions.
Okay, thank you. Yeah, Gisela, would you please go on?
Okay, thank you.
Yeah.
So, and if anyone from our group here wants to make a question, I think we will have time. But let me go for this one. I am Sheikh Traoré from Côte d'Ivoire, and the question is, fossil fuels is a big pollutant. Why most plastics derive it from? We need to divest from fossil fuel. What are the place of plastics, microplastics in this pollutant, and what solutions could we adopt? I don't know who would be interested in responding that.
You mean the question is that, in fact, the bioplastics are now there is a company in the Netherlands, Avantium, who are manufacturing from bio waste. You can make plastics, bioplastics. So I think we should find other ways than that. If this is the question, I don't know if this is the question.
Yes, I think
we need to find another origin, not fossil fuels for making plastic. And one solution is the waste. So, like for instance, agro waste. There are already processes that you can use, forming polylactic acid and other chemicals, which can be used as an artificial or eco-friendly. They call it eco-friendly plastic. So I think we should go in this direction of certainly not using the fuel, the oil for making plastics, and we should get rid of the conventional, especially the single-use plastic, which is still, I think, 40–50% in use worldwide. And so I think for sure I am in favor of this. But of course, these activities will need to be subsidized in a certain time. I know the main companies like Coca-Cola and other ones, big firms are already working on this, because they know that one day they will not be able to use the conventional plastic. Of course, the big companies, they know, but that needs to be, in a way, the authorities and everybody needs to push this, and also the press, the newspapers, and all the social media should go in this direction, I think, to push the use of eco-friendly plastics, waste which is generated everywhere in the developing countries too, and increase the number of at least the proportion of friendly plastic or bioplastic, which is less than 10%, I think, at this moment. So we should go much higher.
Okay, thank you.
Yeah. Do you have questions? I have more questions in the QA. So the time is almost, yeah.
So we still have one question?
For
or two?
Yeah, for other presentations.
Let's see. Well, it's also about microplastics. Let me see. Oh, there is one that I don't know who could respond. It's about treatment. Let me go to this just a minute. So maybe someone that presented on treatment could respond this. Thank you for interesting presentation. When countries are not in the position to upgrade wastewater treatment plants, is there an opportunity for use of smarter treatment process, like increasing sludge age helped to reduce environmental contaminants in the effluents? I think some persons
Yeah, maybe,
presented.
yeah, getting it because the time is, it's timed out. Maybe this question can be just someone can answer in the QA section, so the audience can know the answer. Is that okay?
Okay.
Yeah. So now, thank for the 7 speakers for their excellent presentation in this session, and also thank all of you for your participation. And I also would like to thank the raptors and Zoom supporter recorder for their help for this session. And now I would like to announce the close of this session. And our next session will begin 15 minutes later. So you're welcome to the next session. So thank you. Thank you. Bye-bye.